This is Ahmet Danli from Cold Spring Harbor Lab and Genome Research. This week we have the 86th Cold Spring Harbor Symposium, and the topic this year is genome stability and integrity. Joining me today is Cedric Feschotte from Cornell University at Ithaca. Welcome back to Cold Spring Harbor, and thanks for being here.
Thank you, Ahmet.
Before we start talking about the research, I'd like to ask about your background, where you're from, what inspired you to become a scientist working on transposable elements.
Yeah, so I'm from France. I grew up in France until my PhD, and I moved across the pond about 20 years ago now. And I'm actually a first generation college student, and also the first scientist in my family. So it was not something that was really in the cards for me, I think. And initially, my passion was actually with organisms. So I'm actually a naturalist. Mm-hmm. I like creatures. I like to study creatures, and I was really interested in behavior and interaction and social insects and things like this. This is the kind of scientist I thought I was going to become. However, those plans didn't materialize for various reasons, and I ended up thinking for graduate school, okay, what am I going to do exactly? And I got an idea that genetics and molecular biology was going to be an interesting thing and probably a way to get a job. Mm-hmm. And so I remember still very well when there was a list of projects, to study for graduate school on paper. This is kind of how you pick your project based on the list on a piece of paper. And I see this project about the ecology of the genome and of mobile genetic elements in mosquitoes. The ecology of these elements in the genome. And I was like, wow, that sounds really fascinating. I have no idea what mobile genetic elements are at all. But this is how it started. And really, actually, to this day, this concept of the genome as an ecosystem teeming with transposable elements that interact with each other, interact with their host, and are sort of integrated within the cell is really kind of how we think about things to this day. So anyway, I started my lab in Texas first, and then I moved to Utah, and then now I'm in Ithaca, upstate New York, and we study transposable elements and their impact in a great variety of organisms, mostly vertebrates, but also along the way in insects and plants and all kinds of creatures. So I still have this sort of naturalist eyes, not just on the genome, but also on, just nature. Yeah.
Well, I have to say, I think having been in the transposon field, I really enjoyed following your work. And I think a major strength is your integrative approach using multiple species. And while we're recording this for your talk, and in your abstract, I saw that you're going to talk about constructive aspects of transposable element biology. So you do so many things, I have no idea what you're going to talk about. So could you give us a teaser, please?
Yeah, so there's going to be kind of two parts into the talk. The first part, I'm going to talk about what we've been doing mostly over the last 15 years or so when I started my lab, which is not to study transposable elements per se, actually, to study the remnants of transposable elements, parts, like relic of elements, elements that are no longer mobile, jumping around, but have been sort of co-opted for cellular functions. So I'll start with that and mention a few examples. And basically, the bottom line is we can co-opt parts of transposable elements that could be coding or non-coding. I won't talk about the non-coding part like regulatory sequences, but I will talk about protein coding sequences that have been, repurposed for cellular functions. And really, any activity of the encoded by the elements can be repurposed for cellular functions. And there's many spectacular examples of that now to this day. And I will focus on one type of proteins during the talk, which are GAG-derived proteins. Those encode capsid, which are the defining features of viruses, but also groups of transposable elements called retrotransposons or endogenous retroviruses have these capsid-making proteins that package their RNA and basically traffic these RNAs within the cell and sometimes also between cells. So we can also use them to be infectious. Anyway, so for many other groups have been looking at this kind of co-opted GAG proteins, mammalian genomes, trying to understand what do they do, right? In the context of physiology and development. And there are spectacular examples of that, and one of which we were lucky to collaborate with Jason Shepard at Utah on a protein called ARC,
Yeah, I remember that.
which appears to package its own RNA and traffic this across neurons, potentially at the synapse. And there's a homolog of that in flies as well, called Drosophila ARC. And our only contribution to this work was to found that, in fact, the mammalian ARC and the fly ARC were co-opted independently. It's an example of convergent evolution, where they seem to be doing something similar at the synapse package and that do this RNA trafficking. So we thought, well, it happened, twice. It may have happened more than that. And
That's
so
right.
we and others have found multiple other examples of that. And the question is that I'm going to get into during the talk is, what is predispose these GAG proteins to get co-opted for development or other cellular functions? And what I think is like they have activities that can modulate development when they are part of the transposon. So as an active transposable element, in fact, they already have an activity on development. And the idea is like you create an addiction,
Yep.
and that opens the door on occasion towards like the co-option of the transposon gene. But basically, there's a persistent activity that these elements have, and that can modulate, development. And so that actually makes the elements themselves potentially essential for development. So we are going back to ideas of Barbara McClintock,
Yep.
where transposons are really integrated, and you cannot really extricate them anymore. And so, it's a form of addiction, because probably too much of that is probably not good.
So how widespread is this addiction evolutionarily?
This is really a model at this point, okay?
Yep.
And so we are using the zebrafish to test this. And as you know, we've got into the zebrafish because, first it's somewhat understudied in terms of the genome and transposons in there, yet it's really teeming with transposons. 60% of the fish genome is made of transposons. And what's interesting about this landscape is like they're very young elements, very diverse elements, as opposed to like what you see in mammalian genomes where, mostly elements are dead. Here there's a lot of young elements. So we thought it was a good place to look for the impact of young elements.
And it's a great model for development, right?
Exactly. So that's well established, and that's why a lot of the people use zebrafishes, because it's very tractable experimentally to manipulate and to observe embryos. So we thought this would be a good place to test whether some of the elements, active elements in zebrafish, are actually important for development as active elements. So this was an idea that actually my graduate student, Sylvia Chang, joined the lab, in fact, to test this idea, which I was, honestly, for the first couple of years, I was very skeptical about this. I was like, well, why? Why would they have, they are just, I was raised to the selfish DNA school, right? So the elements are there because they're self-replicating, right? They don't really need to do something to exist. She thought, but maybe you should think about it differently, Think about it as, potential, an opportunity for mutualistic interaction. So if things starts like this, then, and you can see how you can go all the way to the sort of co-option. So maybe there is a gray zone, in a way, between being selfish and purely co-opted. There is a window where the element is actually becoming essential for development. So let's test this. And because zebrafish, is tractable, we can maybe knock down or knock out some of the elements and just look if there is a phenotype in development. And I would present preliminary data, still preliminary, we're working on this, supporting the idea that some of these elements, in fact, these GAG proteins encoded by some endogenous retroviruses in the zebrafish, have an impact on development, in particular, in this case, in mesoderm development. So way outside of the germline, where they normally functions.
Are we talking about this one protein, small number of proteins, a lot of
one,
proteins?
we're going to be looking at one.
Okay.
One specific element called bicari, which was actually described in 1999 and was isolated as an RNA marker of the mesoderm by a group that was trying to find markers of mesoderm. And there's only one paper on this element. It's a very nice paper. They already established the pattern of expression of the element, which is very specific, very tightly controlled. And again, surprising for a transposable element, right? You wouldn't expect them to go into like somite, things like that. Why would they do that? So Sylvia picked this element thinking that, well, maybe it's doing something in mesoderm development where it's expressed. And so she's used a variety of techniques to knock it down and look at the phenotypes, basically, in development. And you see pretty striking phenotypes that are consistent with the role of these product of this element. We're still not sure if it's the GAG, but it only encodes the GAG protein, so it's likely to be the GAG, but it could be the RNA itself. We just don't know yet. But we think it's the GAG, and it seemed to be, modulating cell adhesion processes like this, because the phenotypes are very similar to mutants for like caderins or non-canonical Wnt signaling pathways, and that basically involved defects in cell adhesion and migration and polarity. So we think, because these particles, these viral-like particles, they interact with membranes, and we think that their budding or their assembly may modulate cell adhesion. And so if you remove them, now you have defects in cell adhesion. Basically, it's the working model. It's a work in progress. What I'm going to present here is it's very new. It's very fresh.
That's what Cosmic Hybrid meetings are about, though, right?
Yeah.
When people talk about published work, it's not as exciting or informative in a way.
That's what I was excited to talk about. It doesn't have much to do with the nucleus, by the way. It's like as far as you can be from the nucleus, which is what most people here are interested in. But that's okay. Hopefully there will be something new,
Well, let me ask you something that's more related to, well, not more related to, but another angle. In one of your prior publications, maybe more than one, you were talking about how new transposition is balanced by DNA deletions, and how it might be skewed in some avian species so that the genome is smaller, but there in general tends to be a balance.
Yeah.
So I was curious, does this mean that in a species with an active transposon landscape, do you expect the deletion mechanisms, whatever they may be, to be stronger and more active?
It certainly looks this way. Yeah. In species, yes, it's species. So it was the most striking, at first for us was we looked at this in mammals and birds. We haven't looked actually at species that really delete and turn over DNA much faster, like flies or actually fish. We haven't actually done this. But we looked at this in mammals. In fact, it started because of bats, because bats have the smallest genomes of all mammals. And yet the paradox was, like, when we started looking at the bat genome, it was a very dynamic genome compared to other mammals. We see a lot of elements and young elements that we don't see anywhere else. So it was a paradox. Well, it should be a bigger genome, but it's actually a smaller genome. So it did suggest that there was sort of an adjustment that was being made by which, you would sort of balance the gain of DNA that you have through transposition by some deletion. And by the way, what we found were they are large deletions. So the rate of deletion sort of compensate for the rate of acquisition. It's also true in the mouse compared to like primates. Also mouse has a lot more. The turnover of DNA is much faster than the primate lineage. But the bats was the most extreme, and so that indeed to a point there where the genome shrinks a little bit more.
Yeah.
But overall, there was a striking kind of balance and sort of homeostasis of genome size, both in birds and mammals, where the amount of DNA that you gain through transposition essentially is equated to the amount that you've lost, despite this great variation in the amount of gain. So does that mean that, back to your question, does that mean that how do you modulate the rate of deletion? I don't think you have to modulate, maybe just selection acting to kind of, it suggests that there's an optimum kind of genome size, which, is been kind of, it's an old idea, I think, that, there is constraint on the overall size of the genome because it correlates with cell size. But so this suggests, that's the way we think about it. And by the way, also we think that the proliferation of element would facilitate the removal of the DNA. Okay, so it could be also sort of a neutral kind of process through ectopic recombination between repeats. You have also, more opportunity with young elements to kind of remove DNA in between. So it could be that this fits the circle, right?
I think, the role of repetitive DNA has been kind of underappreciated from that perspective too. We're going to have to see what roles they play in DNA repair, replication. There'll be more to come, it feels like.
Yeah, this is a bit understudied, I think. Well, the idea of patching of double-strand breaks with transposable elements, this has been actually shown a number of times now, way back, in particular in yeast.
Yep.
You can, even though there's relatively few
Yep.
active transposon, their cDNA can be used to patch double-strand breaks. So it's kind of it's not the act of transposition really, it's this whole combination.
Yeah.
But yeah.
Well, probably with the new techniques, it's time to revisit some of these original findings and
I'm sure.
see what they
Yeah,
actually
yeah.
do. I was curious. I think one thing that has been difficult in repetitive elements is to detect their expression, to assemble the genomes. We still don't have the Y chromosome, even in complete genomes. So we definitely rely on technology.
Sure.
So I was curious whether you think for your research there's a particular bottleneck that you think should be overcome, or you think at this point it's about brute force identifying model organisms like zebrafish you mentioned, and just go for sequences and look what happens.
Yeah, I mean, the reads getting longer was really the game changer, right? So when you moved, for us at least, for what we do, looking at the activity, the biochemical activities, the elements in there, transcription, or binding of transcription factors and things like that, or chromatin landscape. When the reads started to get longer than 50 nucleotide, then it became much more mappable within the repetitive fraction of the genome. It was a huge leap. So instead of throwing away all the multi-mapping reads that you could map and not being able to see the activity of the elements, that made a big difference to go to 100. But still, there's some challenges. It depends where you look.
Yep.
Again, if your landscape of repetitive element is a landscape that's made mostly of old elements, they fairly diverge from each other. So for example, in the human genome, we can see pretty well. There are regions of the genome where we can still not see, right?
Yep.
I appreciate that. But then if you move into organisms like mouse, things get more complex, and actually probably zebrafish, we are actually having challenges still with mapping, the locus-specific level, the activity of certain elements, because they're all very young and recalcitrant to mapping.
Yep.
So there's some new sequencing platform I hear that are coming up.
From this week, the news?
Yeah.
It'll be interesting.
Yeah, people are excited about it. Also, long read sequencing as far as transcripts. The long read transcriptomes are really going to be a big game changer with regard to, are already a game changer to looking at repeats and fusion with genes and things like that were still very hard to look at with short reads.
Yep.
So I think we're going to see some exciting things there.
What about manipulating these elements?
Yes.
To supplement the evolutionary findings, and maybe to finally get at the de novo somatic events and disease states.
Yeah.
Have you been doing any pooled CRISPR screens, for example?
Not pooled yet. We've thought about those, but in my lab, we're super lucky to get an amazing postdoc in the lab. And when we were in Utah, Ed Chong, who came to the lab and like, hey, I think it's time now with CRISPR to take out some of those elements. Do the kind of hard experiment, the genetic experiment of just removing them and see if they really are doing something. So he's done that when he came to the lab, and it was amazing. It was a milestone for us. It's not as easy as people often say or write. Even in cell lines and in vivo, it's still tough to do this. And scaling it up to looking at entire family of elements is what we're actually doing now in zebrafish, for instance. It's becoming more possible, certainly. So we can test the impact of the functions of like large family of elements, but it's still pretty tricky. So we haven't done pooled kind of screens, but we've been thinking about those in a cell population,
Yep.
When you delete like one element at a time, type of like perturb-seq.
Exactly, that's what I was going to ask. If you could combine it with the single cell aspect, perturb-seq might be quite interesting.
No, it's on our radar, and I'm sure a lot of people are thinking about this. This would be really, really interesting. And more systematic approach, like taking an entire family of elements, or all the elements that we see like human specific versus those that are.
Yep.
It's this type of like systematic approach. Screen those that may have been like co-opted for functions. Again, we're looking at needles in the haystack typically, So large-scale approaches are the way to go. Yeah.
At least they're becoming more feasible.
Yeah.
Not easy, but feasible.
Yeah.
So I don't know how much time is
I don't know.
yet.
This is not ticking.
We still have 20 minutes apparently, so we can keep talking.
Yeah.
So what are a couple of things that particularly got you excited recently about transposon research?
Well, one I already mentioned is this idea, which I was kind of skeptical about, that elements can actually get intertwined in development to a point that you can no longer remove them. So they've become really important. And, again, back to like McClintock in some way. So that's one. I think another one too is this growing, appreciation that they are that transposable elements can have effects on cell function in ways that we just didn't anticipate or didn't think about. So nucleic acid sensing and the role of like transposon products. They're for better or worse in, so for instance, regulating immune cells. Or immunity or immune responses, modulating these responses. That's been really exciting, I think, So it's not about transposition per se. It's not even about like their replicative abilities or anything, or their regulatory activities. It's just their byproducts. But because they're so abundant, they're such a primary component of genome, and it's kind of like
and the variety.
Yeah, the variety too. And so this is almost surprising, in fact, that people haven't really thought about this. When it comes to like nucleic acids, for example, like cytosolic cDNA is a product of reverse transcription, sometimes aberrant products. It doesn't really matter what the sequence is. It's just the product, the nucleic acids themselves that are being sensed and recognized. So really, it's kind of almost like in some way agnostic to the age of the elements or what they are exactly, So that's this whole like signaling kind of idea. I think that's exciting. I think that's new.
It's fascinating. I think there will be a lot more to come. From the transposable element perspective. So just at the end of the chat, I'd like to ask, you mentioned that you're a first-generation immigrant. And which I think I knew. I'm curious, while the pace of scientific discovery has been very impressive, the communication of science to public has been really lagging. And I was curious, as a scientist, what do you see as what we can do to improve science communication, to increase representation in the next generation, to improve public funding of science, and the value given to science in a way. So we could close with that if you...
Yeah, that's a big topic. It's going to be hard to do in a
2
few
minutes.
Well, I mean, the one thing that comes to my mind is, in terms of communication and getting people engaged, and it's like kids, right? It's like start early. And so going out to like schools, talking to kids about science in ways that they get excited about. Getting then, high school kids in the lab. We try to do that in the summer. Get them a little bit of a feeling of what it is to do science. Can be a game changer for everyone, not just the student. And then undergraduates, I'm very happy where I am now. We're getting just amazing undergrads working in the lab. Also diversifying our undergraduate population is something that we've been, undergraduate researchers, we've been working very actively on. And so, at every stage, if you do a little bit, I think you broaden your base there. And so I think starting early basically is what I think is we got to do.
One of the many things probably we need to do.
Yeah.
All right. Well, it's been a pleasure talking to you. Thank you very much
Likewise.
again. And good luck with your research.
Thank you.
And we'll meet in the next Transposon meeting here, I guess.
For sure.
All right. Thank you.
Bye.
Bye.